But I'm a bio minor and I'm a pre-med student, and I started doing research in the biomedical sciences department last year. And this kind of developed through speaking with the head of that department, Dr. Agarwal, as well as working with my student mentor, Kim Fisher. So, yeah.
The site.
Oh, goodness. Okay. All right, so a little bit of background, because I'm pretty sure most of you had no idea what I just said, or what the title of my presentation means. I'm looking at cytokines and their effects on toll-like receptors in bronchoepithelial cells. So, toll-like receptors are involved in innate immune response, and in total there are 10 different human TLRs that have been identified, and TLRs, and they're named 1 through 10, obviously, but they're named toll-like receptors for their similarity and the conservation of structure and function that's seen between mammals and adult Drosophila flies, and those are called toll receptors. And the toll receptors are involved in antifungal detection in Drosophila flies, and for us, they're part of the innate immune response. So TLRs 1, 2, 4, 5, and 6 are all located on the cell surface, and all the other TLRs are located within the endosome. So these receptors, while they're all membrane-bound, cytokines and anything that's affecting them has to travel through the cell surface to get to them. And an important note for TLR9, it's activated by more than just normal pathogens. It's also activated by bacterial CpG, which I'll explain later on. And so TLRs recognize pathogen-associated molecular patterns, or PAMPs. And these patterns are basically generalized molecular form that is different from host cells. And activation of these leads to transcription of genes that encode for immune response in cells. And I just realized, I think this is the old version of my slides. I emailed myself, so everything might not be on here, and I apologize for that. So the 2 types of cytokines, which are cell-signaling proteins I use, are tumor necrosis factor alpha and interferon gamma. And they're both involved in immune response. And so tumor necrosis factor alpha is produced mainly by macrophages. It's produced by other cell types as well, but macrophages are the ones that produce the most, and it's involved in systemic inflammatory response. As its name suggests, it was discovered in attempts to identify tumor necrosis factors responsible for necrosis of the sarcomere, and it possesses both growth stimulating and growth inhibitory properties. And its role in immune response is varied. It's used for local inflammatory, and it initiates a cascade of cytokines. It increases vascular permeability to recruit macrophages and other lymphocytes to infection. And then interferon gamma is produced most by natural killer cells and natural killer T cells, as well as professional antigen-producing cells and helper T cells. And it's likely to be important in early host defense against infection, but it also, all its properties aren't known, just like for TNF-alpha. And it also induces the creation of antiviral proteins. Okay, for the method behind this process, I use commercial bronchoepithelial cells that can be bought. This line has been continuous for quite a while. And these cells were stimulated with 25 nanograms per milliliter of TNF-alpha, as well as 25 nanograms per milliliter of interferon gamma. And then they were incubated for 24 hours. And this is a lovely photograph of the cells. They're distinctive looking, so they're obviously epithelial cells. And to make sure that there was a control and we could identify what actually happened to the cells, there was a control that was not treated with cytokines, one that was only treated with TNF-alpha, one only treated with IFN-gamma, and then one that was treated with both cytokines to see the effects of it. After the cells were treated, the RNA was extracted using a Creighton kit, and so that uses silica membrane spin columns that removes the DNA but also makes sure that the RNA is still intact, and so it gets the proper genetic material out of the cells. And so then the extracted RNA is quantified, and we figure out the concentration of it, and then it goes through a process of reverse transcriptase and a thermocycler to create complementary DNA from the mRNA. And the reason we do this is the fact that DNA doesn't get affected by the cytokines, but RNA does. And so in order to see the effects it has, we have to find the complement to it in order to run the continued experiment. And messenger RNA in particular is used for protein creation, so that's why we picked that RNA. And then from there, the cDNA product that's formed from the thermocycler is then diluted before being combined with a cytofluorescent dye called cytogreen, and then primers for both TLR 2, 4, 9, as well as GAPTH, which is a housekeeping gene. And GAPTH is already quantified, and all of the information about it is already known, so that's why we're able to compare these primers to it. And so with that, the combination of the DNA products with these primers is analyzed using a quantitative real-time polymerase reaction. And it's important to note that the cytogreen is what's being detected. So it embeds into the DNA, the cDNA, and it's only able to be detected when it's embedded, and it can only bind to double strands. And so when it's released during the qPCR process, then that's how we know that a product's been formed, because the release is detected. And so after that's done, the qPCR product is then run through gel electrophoresis to make sure the right products have formed and there are no primer dimers that have formed. So it makes sure, it's another way of just double checking to make sure everything went through properly. And so the results of this. This is a melt curve from the qPCR, and each of the curves, there's 4 distinctive ones, which shows the 4 distinctive products that were made, one for TLR 2, one for TLR 4, one for TLR 9, and then also a GAPTH curve. And so there are a lot of distinctive curves on there, and it's the temperature that each product is formed at, and as the temperature rises, the DNA strands separate and the cytogreen is released, and so that's what's being detected there, because it loses its fluorescence. And so that's one of the things with real-time PCR, you can detect the products as they're being formed in real time compared to normal PCR, which just detects the products at the end. And so the single temp of each curve indicates the single gene products that are formed. These are the results for TLRs 2 and 4. I was able to run 3 different sets of samples for these that turned out. The standard deviation is a little bit high on some of them, and partially that's due to one of my samples had a much lower concentration of RNA than the others, so that could have had an effect on the product's form. But this just shows the expression and the effects that the cytokines had on these receptors. And in both of them, the combined effect of both cytokines led to an increase in expression, so that's something to note, that these 2 cytokines together have a larger effect than separately. For TLR9, it was a little bit different. The primers didn't work for the 3rd run, and that could be due to a variety of different things. I'm not entirely sure. But because of that, I only had 2 working samples for it. This also shows that the control had a much larger effect than the cytokines, and there are several reasons for that I'll talk about in my conclusions. So yeah. I'm not good at this. Okay. And this is a photograph of the gel electrophoresis that was performed, and this is to double check and make sure the right products were formed. And to explain it a little bit, this was run against a 100 base pair DNA ladder. So the longer the base pair is, the shorter distance it can travel through the gel, since it sort of gets clogged, I guess would be the best way to put it. And so each of the primers used has a number associated with it that says the number of base pairs that should be formed in the product. And so I'm not sure if you can see this. So TLR2 is supposed to form a product that has 139 base pairs, and as you can see, it falls between 200 and 100, and that continues on. TLR9 is pretty distinctive. And then also, you also test that against GAP-DH since it's known and it's supposed to be 94, and that's right around the level. So this sort of concludes that all the proper products were formed. And since I emailed myself the wrong set of slides, I don't have my conclusions listed on here. But one thing that can be concluded, since there was an effect on TLR2, this shows that it is in bronchioepithelial cells, which is to be expected based on other research that's been performed. And also that cytokines TNF-alpha and IFN-gamma do have an increased effect together on TLRs 2 and 4, and that they have a decreased effect compared to not having cytokines on TLR9. And one of the reasons for that, because TLRs are found to be associated with asthma and allergy symptoms in any sort of bronchial situation, I suppose would be the best way to put it. They think it could be an inflammatory response, and so that would increase helper T cells that come in, helper T cells 2, and decrease helper T cells 1. And so they're trying to find effects on that, and so the way that would work in terms of research with TLRs is the fact that because TLR9 doesn't have a large reception to these cytokines, it would possibly be because that there is a large amount of these cytokines already found within the cell. And since TLRs 2 and 4 are located on the outside of the cell while TLR9 is inside the cell, because of that, that's why it might not have a reaction to the cytokines. So it could be because it's decreased activation of it. And so the thing they're looking at is the fact that there's unmyelinated cytosine phosphate guanine, which is the CPG I mentioned earlier. That does affect TLR9s, but it doesn't affect TLR2 or 4. And it's found in microbial viral DNA sequences, and because it can activate TLR9, it could be used as an inhibitor. That would cause TLR9 to perhaps respond better in immune response. So that's one thing that can be concluded on it. If I were to continue this research, obviously I'd want to run a few more tests with these samples just to make sure that the results I have are proper. Also, I would want to replace the primers used for TLR9 just to make sure that everything is still conclusive. I suppose another area I could look at would be the proteins that are formed and do protein analysis, since this should have an effect on the proteins formed as well. So that's about it. I really want to thank the Biomedical Science Department for allowing me to do research, Dr. Agarwal, and particularly my mentor, Kim. And are there any questions? I realize that was a lot, and probably a lot of this vocabulary most people aren't familiar with, but